This meeting is being recorded. Great. So I'm the Education Director with NOFA-Mass. I run our technical assistance services at NOFA-Mass as well, and our soil health projects. I have the pleasure of working with Nagisa, as well as Dina and Jeremy at NOFA Connecticut on a Conservation Innovation Grant from the USDA NRCS. The Conservation Innovation Grant, or SIG grant as we sometimes call them, is a 3-year project to help organic farmers understand how to reduce tillage on their farms. So we'll talk a little bit more in this presentation about soil health practices. But one of the aspects of this project is that we offer soil health testing and observation for those farms to help them track soil health outcomes or sort of indicators of how well their management practices on their farms are building soil health. The tests that we use are a version of the NOFA-Mass carbon proxy tests, which are a set of tests that were developed by Jack Kittredge at NOFA-Mass, who's our longtime policy director. He edits The Natural Farmer. He was one of the early people involved in NOFA's organic certification programs. He's been an important mentor for me. And so he did some research on soil carbon and soil health and developed a set of tests that draw from organizations like Cornell and NRCS field protocols for soil health assessment and other organizations like the Soil Carbon Coalition. So sort of different methods that different organizations and agencies are using to evaluate soil health and track soil health. We have since revised the protocols over the winter, and we're just about wrapping up that process right now, to bring those protocols into alignment and make some adjustments so that they're very similar in many ways to the current NRCS field soil health assessment protocols, which were just released this year. Brandon Smith is a leader at NRCS in soil health and is doing a lot of great work, and so they came up with a new set of soil health protocols this year. So we've adjusted our soil health tests a little bit to make them similar to those protocols. So why do we call them the carbon proxy tests? They're called the carbon proxy tests because soil health and soil life and soil biodiversity and soil carbon are all proxies for each other. The amount of life that is present in the soil is equivalent or highly correlated with the amount of carbon in the soil because most soils, especially in the Northeast, contain very little geological carbon. Most of the carbon in the soil is from the living things in the system. And soil health can be observed through some really basic techniques, and land managers can really learn to understand their soil health through learning to observe different factors of the soil. So that's what we're going to be talking about today. Carbon is really hard to directly measure except through maybe loss on ignition, which is one of the ways that labs will give you a soil organic matter reading. But in terms of in the field, it's really hard to directly measure. So these holistic methods that involve observing the soil ecology or indicators of the soil ecology are just a really good way to kind of get a sense of how much carbon your soil is supporting and drawing down out of the atmosphere. So should I pause for any questions about that, Nagisa, or should I just keep going?
I think you should just keep going for a little while.
Okay, all right.
I haven't seen any questions yet.
So do you want to try to play that first video at this point and switch to your screen? I guess not. Okay. So the first thing you do, and we have some longer form videos. Nagisa sent out a folder, and in the email there's a series of videos you can watch. The videos that are embedded in this presentation are really just
sorry.
Kind of,
there's a bit of a delay.
Oh yeah, I know. I'm sorry.
So I am fixing to show it. Do you want me to show the first video?
Yeah, sure. That would be great. Let's see if it works. And yes, there is a delay. So I'm sorry about that.
Okay.
It's just going to be a challenge we have to deal with.
Okay.
Hey, bud. I'm the Education Director with NOFA.
Oh.
We can skip that part. Caro Rozell. I'm the Education Director with NOFA Mass, Northeast Organic Farming Association, Massachusetts chapter, and I am also our...
Oh, having a little trouble here. Sorry, guys.
I'm Caro Rozell. I'm the Education Director with NOFA Mass, Northeast Organic Farming Association, Massachusetts chapter, and I am also our Soil Carbon Program Coordinator and a Soil Health Consultant. And I am out here today at Massaro Community. If it's not working, you guys, so we can just switch back. It seems like it's stopping. Okay.
Yeah.
Switch back.
Yep.
Okay, it was worth a shot. All right, sorry about that, folks. We're trying to learn how we can show videos through Zoom, but it's really not that easy. I am going to try to play a short video here that's embedded in this PowerPoint. Hopefully you can see that. The series of videos that Nagisa sent out in that email are actually of Massaro Farm, which is one of the farms in our CIG project. And it just gives a better idea of the test because it was filmed during the growing season. This was actually filmed late this winter, early spring. But what I'm describing here in this slide and in this video is just sort of giving an idea about finding your test location. There's really two ways to use a carbon proxy test or to use soil health assessment. One way is to compare two different management practices on different fields in your farm, or in a pasture or undisturbed meadow versus your growing area. Another way is to track change over time. If you want to track change over time to see how your management practices are impacting your soil health over time, it's really important to choose a permanent location where you can go or a test location that you can measure to from a permanent landmark, so you can go back and easily test that same spot repeatedly. So I have a market garden, so I don't have a lot of land. I chose a spot as an example spot for this demonstration from my chicken coop, or the corner of my chicken run, which is a relatively permanent landmark. I don't want to, use my caterpillar tunnel or the cold frame in this vicinity because both of those things are movable. I move my caterpillar tunnel every year. So I'm just showing here that I've used a ground staple to fix my surveyor's tape, and then I've measured to the observation hoop, which is just a simple 30 centimeter hoop. And what I do if I'm taking a change over time measure is I use that spot as my permanent test location, measure the amount of feet to that spot from a permanent landmark using my cell phone compass. You can also just use a regular compass. However, if you are taking a series of tests that you're just interested in sort of comparing different management practices across your farm or different soil types, then you don't need to do this. This is really a step that's really only needed if you're going to be tracking change over time. So the next test is surface biology. The observation hoop is just sort of to help you focus on one area and estimate percentages within that area of, we're interested in three things. We're interested in the percent of soil that's covered, period, the percentage of the soil that's covered in living plants, and the percentage of bare soil that's visible. So with this observation hoop, a cool trick is if you get a 30 centimeter hoop, one hand spread out wide like this is about 6% of a 30 centimeter hoop area. My hands are a little small, so I usually estimate 5% for my hands. And then your fist on this side, like this, is about 4%, and then your thumb is about 1/3 of a percent. So you can kind of use those handy measurements if you're using a 30 centimeter hoop to track this data. This is basically a way of quantifying your soil coverage. A lot of us think, about cover cropping and keeping the soil covered is really essential to soil health. But you might not have a good sense of how well you're actually keeping your soil covered at different times throughout the year. So one way you could use this test is just to actually take your hoop out, throw it on your field in the spring, right after you planted your cover crops, a month into the growing season, in the height of growing season, after tillage, and again with your fall cover crops, and just kind of take records for yourself about, all right, what percentage of my soil is actually covered with either plant residue, mulch, or living plants. So the reason that this is important for soil carbon and soil life is that the fact of plants of your soil being covered in living plants means that photosynthesis is happening. There's actually companies that are starting to do carbon quantification for carbon offsets for farmers, and they're actually doing a lot of that through satellite. So we're talking about big Midwestern kind of monocrop farms where they do maybe like one cash crop and one cover crop. But there's actually models for estimating soil carbon sequestration using satellite data that just gives days in living cover. But days in living cover is a really important metric for thinking about how well you're doing as a soil steward. The other aspect of this, of course, is soil cover with residue or mulch. Keeping the soil covered is really important for supporting your soil biology, keeping good moisture levels, creating an ideal habitat for your soil microbiology. And then, of course, we want to know what percentage is bare soil. The other aspect of this is to just write down the number of species that you see. So when I do this test as a technician, I record every species that I can identify and the number of different species that I saw. So species biodiversity is also really important for soil health because different soil microbes form different relationships with different plant families. So your above ground biodiversity supports your below ground biodiversity. And generally the more biodiversity you have above ground, the more you'll have below ground, and the more biodiversity you have below ground, the more total life you can support because more complex ecosystems support more life overall. So again, looking at total soil coverage, amount of living plants, and then species biodiversity as well. There's a lot of great research out there if you're interested in really digging in on the way that it's not just different plant families, but also different root sizes, shapes, textures that impact total carbon sequestration in the soil. So I think there's a question about that.
No, I don't think there are. I think you can keep going. We can try another video if you like.
I have a little video in this one if that works,
okay.
Or we can try the video if you'd like, either way.
Okay, well you can talk about it a little and then I'll go ahead and pull up the video.
Great. So the next part of the tests we actually dig into the soil. So we've recorded our surface observations, and now what we want to do is dig a hole and start to observe what's happening in the soil. So for this, all you really need is a shovel, a trowel, and a measuring tape. It's a good idea to always remove a cubic foot of soil so that you have a consistent hole if you're tracking change over time. And especially because we're going to be removing the soil and putting it in a bin to count our earthworms. The number of earthworms per square foot or cubic foot is a great way to sort of evaluate your soil health and compare it. You always want to take the earthworm count test at the same time every year because earthworm population is really impacted by weather conditions, temperature, moisture conditions. So as much as you can take that in basically the same week or at least the same month every year for comparison over time, that's great. And if you're doing comparisons of earthworms across management practices, just do them all on the same day if you can. In addition to counting the earthworms, you'll be counting the biopores in the surface of the hole. What that means is those little holes that you see running through your soil that are the passageways for either roots or earthworms. Oftentimes when you're looking at a subsoil that has biopores in it, you'll notice that the biopores are actually lined with darker soil because the roots and the earthworms are actually bringing biology down into that lighter subsoil and spreading the soil ecology deeper into the soil. It's really cool. So we want to count the biopores that we see on the walls of the hole and just record that to know, okay, so what's the relative sort of activity of the biology, at least in terms of roots and earthworms. We also track the number of, or the deepest root. It's a good idea to record the deepest root that you see present, and notice if any roots are J-rooting or turning sideways suddenly. That can be an indication of compaction or of a seasonal high water table or of some kind of issue in the soil that the roots are avoiding. If you see any balled up roots, they're like stopping and kind of just like balling up, that's a bad sign. So you're looking for how are the roots behaving? Are they moving straight down into the soil? Are they able to grow straight down, or are they kind of turning sideways? What's the deepest root, and where do the majority of the roots terminate? For looking at root depth, it's a good idea to do this when there's a mature cover crop or crop in place, so you can really get an idea of how deep the roots are going, not just how deep they've gotten so far in the season.
Oh my God.
What?
Okay. Shall I give it a shot?
Yeah, let me just get a couple more points across. The other thing we're looking at for is topsoil depth, so the depth to the color change. And then we'll talk about aggregation in the next slide. Let's see if we can make that video work.
Okay.
Dig a hole. We're going to do a number of things with this hole, but it's important that we want to dig a cubic foot of the soil and displace it into a bin or a tarp. I used to carry a tarp around, but I've just moved on to kind of really preferring having a bin. The reason for that is we're going to be looking for earthworms in that bin, and I just find it a little easier as I'm getting to the end of my cubic foot of soil to sort through it from a bin. In this particular hole, the majority of the roots seem to terminate at about 5 inches, which is really good for springtime. And we have at least one root that goes all the way to 12 inches, and I'm seeing a couple other ones that are at about 10 or 11 inches, so that's really good. So it doesn't appear that any of the roots are sort of hitting compaction points. None of the roots are abruptly turning sideways. None of them appear to be balling up. So we have good root behavior. The next thing we're looking for are biopores, or sort of channels in the roots left by earthworms and roots. And at this point, we just count what we see in terms of biopores in the hole walls. I'm seeing about 3 visible biopores on average per wall. So about 12 in the hole. Just double checking that. Fewer on this wall, so maybe we'll say 10. Yeah. The other thing we're looking for in the hole is the depth of topsoil. So what we're looking for is a color change. On a farm that's tilled regularly, I usually see a pretty bright line of dark colored topsoil, light colored subsoil, usually at around 9 inches, typically, and that's the depth of the plowing on that farm. And they develop plow pans, so you just see a really large distinction.
Not good.
On a farm like Massaro Farm that has been using no-till practices, you often see a much fuzzier gradation, a marbled appearance, and we clearly see that on this site. The color change from the dark topsoil to the lighter subsoil is much more gradual, and like I said, marbled is the best way to describe it. So you see here on this slice with the painter's trowel, which is a really important tool for your soil health assessment. Painter's trowels are invaluable. We have this nice dark topsoil, but it doesn't transition abruptly to the lighter colored subsoil, and that to me is a sign that the merging of the subsoil and topsoil is happening a lot through root and worm and other biological activity, and not through just plowing and turning over one layer of the soil. Another aspect of the soil system that we're looking at when we do the digging a hole test is root sheathing. So this is basically the strength of the relationship between the plants that are growing on site and the soil community. You can see here that these roots are very tightly sheathed in soil, so that you can't see very much of the primary root through the soil clinging to it. Grass is one of the better, I just went and grabbed this clump of grass because grass is one of the better plant families to test this on. But it's a sign that there's really strong relationship between the soil biology and the plant. So there's a lot of exchange happening in the form of carbohydrates and other components of the root exudate that is causing the ecology of the rhizosphere to have a strong connection with the plant. Other plant families, well, that relationship is going to look a little different. Like typically, as most growers know, onions don't tend to, their roots don't tend to sheath as much. Here we have some chickweed that you can see a lot of the root through it, but at the same time, there's a lot of aggregation growing off of these roots. But generally, if you have a grass present, I like to stick with a grass to test the root sheathing. I just go find one near the test location if there's not one right in the hoop. But this is really, really nice sheathing here. If you saw just kind of, if it was more similar to the onion root and you could see a lot of the root through the soil, that's less good sheathing. That's not as good sheathing. So you're really looking for that really nice thick coating of soil on the root.
All right. So with that, I'm going to hand it back to Caro to do the next slide.
Great. So can you see my screen, or do I need to reshare?
You need to reshare.
Okay, thanks. We'll get the hang of this eventually. So any questions about digging a hole? Oh, I see a question on chat. If that's all right, I'll just send it. Do you use a quantitative scale for things like sheathing or just a verbal note? Not sure what a verbal note means, but it's really just, a lot of these things are just experience-based. So if you get out there and just do this regularly, and also there's a lot of really great photo guides on NRCS's publications, you'll start to, the best way though is to really get to know your own land and to sort of pull up grass in different spots on your farm, maybe like a really tilled area or highly disturbed area versus a pasture area, and you'll start to sort of see those connections and be able to compare across different examples on your own land. So yeah, there are photo guides, but a lot of it's just building up a personal observation inventory.
Madeline's asking, what's the easiest way to dig a hole?
Oh, yeah. So I usually carry just a regular spade. I know a lot of folks are really fans of like the edging spade because it gives you that nice, the kind that you like professional landscapers use. You can get a nice clean edge with that. I'm a farmer, so I usually tend to have regular spades on hand. So I carry a painter's trowel, which is that nice flat diamond shape but flat trowel, which lets me get a nice smooth cubic foot because I'm really going for that nice precise cubic foot as much as possible. So I first spade it out, put all the soil into my rubber bin, and then I use my measuring tape and my painter's trowel to get the nice, cubic foot. All right. So, any other questions about digging a hole? We already talked about earthworm count. Just a note, some people say, why are we using earthworms as an indicator for soil health when they're not native to the United States? And what I would just say is, they are they like the same conditions that are the conditions of soil health. So like a soil that's rich in microbiological activity, that has lots of organic matter, that's evenly moist but well drained. So they're sort of just an indicator of a healthy system. I kind of think of them as the whales of the soil. They kind of move through the soil, taking it in and actually digesting a lot of the microbes out of it, and feeding behind them with the microbes that that are beneficial for the soil, and they're sort of climax predators. Is that is that the right corollary? So kind of like whales. If an ocean's supporting a lot of whales, it means that there's a lot of other ecology in that soil, in that ocean. Same thing with earthworms. So again, take this test the same time every year. This is one of the most impacted by time of year. All right, so soil texture and aggregation. This is probably the hardest test. This is a test, like we were just talking about, building up your own inventory of observations about your land. This one takes a while to learn. It's a good idea to go and also look at other people's soil and kind of handle it. There's also a really great resource, such as the NRCS's field guide for describing and sampling soils. You can get a PDF of that on NRCS's website. And it will describe to you how to classify aggregates. We'll also be following up this presentation with an email to folks with our NOFA guide, which is a little simplified version of the NRCS guide. But basically what we're looking for here is we're looking to understand the soil structure. So in this slide, you can sort of see a wide range of soil structures. Toward the top of the slide, you can see some kind of more loose, unaggregated soil where we're getting a little lighter toward the subsoil. We're actually looking at a profile of my soil in reverse. So the top is actually the lower part of the soil. Getting toward the top, we see a lot of macro aggregates. So large clusters of aggregates. We see roots running through them from my early season field pea cover crop, which had just started to germinate at this point in the season when I took this photo. And then we're seeing a bunch of different types of aggregates in between. What we're looking for in aggregates is sort of a cottage cheese texture. That's a granular aggregate. In this image, you see some granular aggregation. You also see some kind of blockier aggregates. If you look toward the top of the slide, near to the topsoil change there, to subsoil where it's a little lighter, we see some blocky, subangular blocky aggregates that are irregularly shaped. But then in other parts of the slide, you can see that there's more of like the cottage cheese-shaped aggregates. And then those macro aggregates, the nice thing about, what's interesting about those is if we break those apart, they just fall into more aggregates. The other kinds of soil we might see, if the soil were not quite as healthy, would be kind of a playground sand kind of texture, where the soil just is completely unaggregated. It looks just, it's not, there's no structure to it at all. It would just slip through your fingers. And then we might also see platy or cloddy formations of soil, which would be clods of soil that are really compressed, like if they were really dry and you were to squeeze them, they'd just pop into dust. That's just a clod. It can also be described as platy. And that's an indicator of, that's massive soil it's not aggregated, it's just compressed together. So those are not aggregates. But here we see a range of different types of aggregates. I also, if you're looking at, can you see my video? I have do
you want me to play the video?
Oh, no, I'm trying to show on my speaker video. Can you see this?
No.
No? Okay. So one of the ways to tell that you have an aggregate is that it has a lot of surface area. So it has a lot of little tiny pores. It might have roots running through it. It might have little tiny biopores in it. It might have large biopores in it. You might see like an earthworm tunnel going through it. If you break the soil apart and it has really smooth sides, that's usually an indication that that's a platy or cloddy structure. So you're really looking for something that's has a lot of surface area, is sort of like really finely textured, and when it breaks apart, it just breaks apart in these really uneven, high surface area forms. Does that make sense?
Yeah.
Okay, great. So aggregation, really important, one of the most important soil health indicators. Soils that are well aggregated will absorb water more readily, will hold more water. Plant roots can move through them more quickly. They're correlated with lower compaction, and they're really a good indicator of a good biodiverse soil.
Okay. So let's go ahead and try and look at the video.
About the soil texture?
The aggregation one.
Okay, I think I just said most of what's in that video. We might, for time-wise,
Right.
we might want to skip that one and go to the next one.
Okay.
All right.
Oh, while you're going to the next one, Madeline is saying that she put her earthworms in a compost bin and they died. And how does she keep them alive? I think she probably shouldn't have put them in the compost bin, but can you say any more than that?
Yeah, is the compost bin, can I ask Madeline, is the compost bin the kind that's doesn't have contact with the earth? Is it like its own separate contained bin? Is she answering?
No, she's not. She might be having trouble.
Yeah, so what I'm a huge advocate of compost bin structures that have contact with the soil below them, because you really want your earthworms to be able to migrate in and out of your bin, because if there's a condition in that compost, like it's too acidic or it's too dry or it's too wet, it doesn't have like enough bedding or carbon, then your earthworms, if they can't get away from that, they'll die. So you really want to be thinking, like earthworms like high fiber compost. They like to have a lot of carbon materials present. So you want to give them the option of migrating out of your compost bin and into the soil. Also, those compost bins that are separate from the ground can get really hot, which will kill your worms, and they can just get really anaerobic. I mean, they're not good environments for worms. So if you want to have a good environment for worms, which I do, I use a lot of like wood chips and newspaper and cardboard and leaves in my compost pile. I try to keep it high on the carbon range, and that really invites worms, and I keep it connected to the soil so the earthworms can go down into the earth underneath.
Thanks.
All right, so why don't we try playing the flake test video instead of the slide for this one?
All right.
So the sleep test is basically a test of how well the soil holds up to water infiltration or sort of like water submersion. The amount of sort of biological glues in the soil holding the soil particles together create kind of water resistance for the soil structure. So what we're going to do is take these two samples. You want about a golf ball size of each. They should be about the same amount. And we're going to place them in these ball jars of water that have little baskets made out of hardware cloth in them. And we're just going to place them into baskets and see what happens. So we're looking at really two factors here. We're looking at how much of the clod of soil remains in the basket and doesn't fall through the hardware cloth. And then also we're looking at how cloudy is the water. And you can certainly do this test in a way that takes a quantification by recording the percentage of the soil that stays in the basket after a minute compared to falls down to the bottom. And for the water murkiness, we're actually looking at different field turbidity tests that are used by water evaluators, like folks who do rivershed or watershed river analysis, just to find a good quantification for this. But if you're just a farmer gardener kind of wanting to understand what's happening in your soil, a good way to do it is just to do this, to compare an undisturbed part of your soil with a nearby disturbed part of the soil. So here you can see that there's really not a whole lot of difference between these two. So the field edge sample looks like it's maybe shed a tiny bit less of its mass down to the bottom, and I'm seeing maybe a tiny bit clearer, maybe a little bit more murkiness in the infield sample, but my guess is that because Steve has been using these really great tillage reduction methods and you've got a really good cover crop in place and lots of soil cover, we have a good, strong amount of fungal and bacterial glues in the soil holding particles together. So we're not really seeing a huge difference between these two samples, which is really a really nice indicator of health.
Okay.
Great. So on that note about compost and worms, this book is called Compost, the Natural Way to Make Food for Your Garden. It's just a really simple book with like a lot of beautiful pictures. And it describes the high fiber composting method that I just talked about. I highly recommend this book. It's a lot of pictures. It's really a beautiful little book and has a lot of good info about composting.
You know, that's an indication of how extreme you're getting. You're telling us that a book of worms is really beautiful.
I really do. So I wanted to since we're talking about the slake test right now, I'm happy to take questions about that, but essentially what we just saw is the two clods, you know, one from the field edge that's relatively undisturbed and the one from in the field actually were pretty similar. Often when I do slake tests, the test from the area that's in production will make cloudy water, and a lot of the aggregate or the clod of soil will fall through the basket to the bottom of the jar, while the undisturbed edge will usually the water will stay relatively clear and it'll mostly stay in the basket. That's typically what you would find, but on no-till farms, often you'll find a lot more similarity between the two jars, which is what we found with Steve's farm. So I want to take a second to talk about glomalin, which is the fungal glue, which is partly responsible for that ability of the soil to hold together and resist water, the impact of water breaking it apart. Glomalin is about 30 to 40% carbon, and it is thought to be only created by arbuscular mycorrhizal fungi. And it is like we said, it's a carbon-rich compound. It's essentially a water-resistant compound. And it's used in a variety of ways by the fungi. It's used to as a protective coating for the fungi. It ends up, holding the soil together. Whether or not the fungi is creating soil structure intentionally is a question for philosophers, but it does have the effect of giving the soil structure. Another cool way that it's used is that it actually excludes other bacteria from the bacteria that the fungus is specifically collaborating with. So if you just Google fungal superhighways, you can find some really neat research and studies and information about the ways that just as plants are forming associations with arbuscular mycorrhizal fungi, those are the specific fungi that work with plants to take their soil carbon and trade carbohydrates for nutrients and water in the soil. Those arbuscular mycorrhizal fungi then form similar associations with specific bacteria. So the plant is photosynthesizing, has a relationship with these arbuscular mycorrhizal fungi, which wrap around the root and actually exchange for carbohydrates or sugars from the plant for nutrients from the soil that the fungi is providing to the plant through its roots. But the fungi is in many cases getting those nutrients from bacteria, specific bacteria that it works with in the soil. And the bacteria are, bacterially, enzymatically weathering the parent rock material to extract. They're mining those nutrients. And then they're giving those nutrients to the fungus for some of the sugar that the fungus just got from the plant. And the fungi use these glomalin-coated tubes to allow the bacteria that they work with, their worker bacteria, to move along those superhighways of glomalin to get to different places in the soil where the fungi wants them to go to mine these materials for the plant. So it's this really cool underground infrastructure, and glomalin is a big part of that story. It also persists in the soil for a long time, 7 to 42 years, according to different research and management practices, which means that it's one of the main drivers of soil carbon sequestration through plant-fungal relationships. And there's a really cool study. If any of you were at our summer conference last year, we had Dr. Chris Nichols, who's a former USDA scientist, and she's also worked for Rodale Institute, the chief scientist for Rodale Institute. She did this really interesting study on Glomalin, where they artificially changed the atmospheres in these atmosphere-controlled bubbles and raised the CO2 level in those atmospheres, and looked at how the soil biology responded. And what they found is that when they raised the CO2 levels to 670 parts per million, which is a really scary amount to think about, the fungal hyphae, right, which is the underground body of the fungus, grew 3 times as long as the control at current CO2 levels, and produced 5 times as much glomalin as those plants that were growing in today's, well, at the time the study was 670, now we're at 410. But that's really interesting to think about. So that amount of an increase in carbon dioxide in the atmosphere, in the controlled atmosphere, produced 5 times as much glomalin as a fungal response, which is really interesting when you think about that being one of the main drivers of soil carbon sequestration. So if the fungi are responding to climate change, elevated CO2 levels, by making more glomalin, that's one of maybe the buffer feedback mechanisms, the mechanisms that are reducing the feedback of climate change. So I find that really comforting as a research study, and I think it emphasizes the importance of fungi in our system. And this brings us back to why it's so important to reduce soil disturbance, because when we till the soil, what we're doing is we're promoting a bacterial-rich soil, and we're demoting the presence of fungi in that soil, because fungi exist in the soil as these fine hyphal strands. And tillage is just fundamentally antagonistic to that physiology, that hyphal strand physiology in the soil. And when we break apart those hyphae, when we knock back our buscular mycorrhizal fungi population, we're eliminating that middleman between the plant and the specific families of bacteria that symbiotically associate with those mycorrhizal fungi. I mean, we're certainly supporting populations of bacteria that work directly with plants, but you have a much more biodiverse soil environment when you have arbuscular mycorrhizal present, and you're also doing things like putting glomalin into the soil and creating good soil structure. So this is just why, when we think about glomalin, we think about hyphae, we think about the role of mycorrhizal fungi in the system, this is why it's important to be reducing tillage. Of course, tillage also does other things like, oxidizes soil carbon and causes the release of nutrients and reduces soil structure and reduces water holding capacity and all those other things. But just think about your fungi when you're disturbing the soil. Any questions about that?
Doesn't look like it. Oh, somebody wanted to know, can you repeat the name of the book one more time?
Oh yeah, Compost, the natural way to make food for your garden. This is probably looking backwards to you because I think the videos are mirrored, but it's published by DK Publishers.
Okay, I just typed it into the chat.
Great. And if you're looking for a good book about mycorrhizal fungi, Michael Phillips, who's a longtime NOFA person, an orchardist, has a book called Mycorrhizal Planet by Michael Phillips. Really recommend that book. So let's talk about infiltration. Do you want to try this as a video, or do you want me to just play it from my screen, Nagisa?
I'll go for it. Okay, hang on just a second.
Great. So while Nagisa's doing that, oh, here we go.
The infiltration test is a test that measures how quickly water infiltrates into the soil. Different aspects of the soil structure, how well aggregated it is, how good the sort of capillary action is from root channels and earthworm channels into the soil. All of those things impact it, whether there's surface crusting. So it's a really good measure of soil health. Typically we see anywhere from 4.5 seconds to up to 45 minutes for this test. It's really wide ranging. We'd like to see things under 20 seconds. Ideally, under 10 seconds is really good. So the way to do this test is you bang in a cylinder. This is just a basic sewer, 6-inch sewer pipe, plastic sewer pipe that's been sharpened, cut to 6 inches and sharpened on one end. Use a piece of scrap lumber to pound it in with a mallet. And then we use just basic cling wrap covering the surface to allow us to pour the water in and then start the infiltration all at once. For a 6-inch cylinder, it's 444 milliliters of water that we use. So I just carry a jar that has that amount marked out on it, and I pour it into that container on site and then into the cylinder with the plastic on it. Pull the plastic off and start my cell phone stopwatch at the same time. Any stopwatch will do. Since I use a phone for so many aspects of these tests, it's just the easiest thing. And so this test was, we got 17 seconds for infiltration, which was a good reading.
Cool. What's a bad reading?
I have definitely had a farm that I was consulting for where I started it, and 45 minutes later, the level had barely moved.
Well, that's terrible.
I just packed up and left. I just wrote down 45 minutes plus. I had to leave, you know, because it was getting dark. It was rough. Yeah, so it really ranges, but you know, under 20 for clay soils and under 10 seconds for sandy soils is what we like to see. I keep doing this from the beginning. I'm sorry. So that's infiltration tests. Any questions about that? I think one of the things that's important to understand about the infiltration test is that when you're thinking about how quickly your rainfall is able to infiltrate into your soil, what we're actually thinking about is like how much rain do you need to get before on your soil you get overland flooding, which can lead to erosion, and then downstream impacts. So sometimes, you know, there's a lot of news about flooding increasing, and I think that definitely we're seeing more precipitous rainfall, but I think another thing that maybe isn't considered as much as it should be is that with as we degrade our soils, especially in agricultural regions, we're degrading the ability of the farmlands to actually absorb rainfall as it's coming in, which leads to more flooding. So I think it's a really important large-scale externality that can be a result of soil degradation, and increasing soil health, I think, can reduce flooding issues for agricultural regions.
Cool.
So soil hardness. This is a test that does involve a tool that we purchased, an Agritronics penetrometer, which you can see here, which is you know, a $170 tool. You can certainly do just an observational test on your own with one of those little field marker flags, just poking it into the soil and seeing how far you can go before it bends. And that would be something you could use to compare different fields and compare field edges with your fields to see where your compaction is. But the nice thing about this tool, you can see I'm pushing it down, and it's actually giving me a reading of the pounds per square inch of pressure that it takes for me to push the point of this tool into the soil. So it's a nice quantified reading. So when we do a technical visit for a farm, we actually give them a report that gives their average PSI across 3 readings for every 3 inches to 24 inches. 300 PSI is the pressure at which basically all crop roots can't really penetrate. And then there's a nice handy table here that comes with your penetrometer that sort of shows for the average crop roots what percentage can penetrate at different levels of PSI, different PSI readings. So you can see that it's a pretty quick transition from most plant roots being able to penetrate to very few being able to penetrate within that range of 50 to 300. So on tilled farms, I do tend to find a compaction that starts at anywhere between 9 and 15 inches. I have found compaction levels at even as shallow as 7 inches on some farms that have a history of tillage. But on no-till farms, I tend to find much lower PSI. I have two extremes here in the slideshow. There's a tilled grain farm that I've tested and a no-till vegetable farm. These are two, kind of on the far sides of the spectrum that I've seen. This no-till vegetable farm has particularly light soil, but yeah, it is typical that we find less compaction on no-till farms.
Great.
Yeah, so if you are concerned about soil compaction, I do recommend getting one of these tools and exploring in your own field, or calling a local NRCS representative who might be willing to come out and do this for you. So I think the last test I'm going to talk about today is also something you would buy a kit for. This is the active carbon test. You can get it from Soil One. I think it's just soil1.com. And this is a test. It's potassium permanganate. It's a reagent. It's dark purple when it comes out of the bottle. And what you do is you mix it with your with 5 grams of your soil. There's instructions that come with the kit if you're interested in getting it. And you shake it. You have to shake it for 3 minutes. And at the end of that 3 minutes, you let it sit for 10 minutes. And then you look at your vial. So you've mixed 2 milliliters of your reagent in with your soil and 20 milliliters of water. Shake it up in a clear vial. Let it sit for 10 minutes. And after 10 minutes, you will have anywhere from this dark purple that's shown in the slide to all the way to clear. And what that's measuring is the oxidizable carbon or the exchangeable carbon, or what's also called the labile carbon. That's basically the carbon that's being actively exchanged. So not humic compounds, not like chunks of wood, but the sort of the carbohydrates and the other exchangeable forms of carbon. What's interesting, so the clear means you have a lot of active carbon, and the red or purple would be not much active carbon. What's nice about this is you can often using this test detect impacts of management practices long before you'll see a result in your soil organic matter reading from your lab. And if you take this regularly, you can actually predict trends in your soil organic matter. So if you're concerned about if you're starting to till up a new part of your farm and you want to see how that impacts your active carbon, you could take an active carbon test before you, while there's a pasture growing there, and then if you till it up and turn it into a vegetable field, take it again, see what happened. Usually you'll see a spike in exchangeable carbon right after tillage because we've just killed a lot of soil life and some of it's actively being decomposed. But then like a month after tillage, you might see a sudden drop in your active carbon. So it's also a nice test to sort of compare soil cover. If you have a lot of soil cover, a good thick stand of a cover crop, you'll probably see a higher active carbon reading than a similar part of, a different part of that field where maybe you didn't get good germination and you had bare soil. So it's really a nice way to sort of just see what's happening with your carbon levels in your soil. So that's Soil One. You can order a little field kit and use that on your farm as just a way of kind of taking the temperature of.
It's $19.
Right. So here's a question. Yeah. So just so everyone, I'm going to remind everyone that each of the tests has a video with it. And each video, you were sent a link in the invite to today's webinar. So feel free to go online. It's on the NOFA New Jersey YouTube channel, but it's not publicly listed. So you needed to have signed up for it to get the link. Okay?
And also in that set of links is also a link to some soil microscopy, a little soil microscopy demo by Monique Bosch from Connecticut NOFA. She's a partner on this project, and if you're interested in checking that out too, I didn't talk about microscopy today because it's not my area, but I think that'll be an interesting little video to view.
Great.
Any questions about what we just talked about?
I think we might be good.
Great.
I don't see any questions here. Does anybody have any questions? You know what? Here's what I'll do. I'll unmute everyone so that if you do have a question, you can ask it. Okay? So, but when I unmute, please do us a favor and try not to make a lot of noise or remute yourself if you know you're in a noisy place. Okay? So here we go. Okay. Anybody have any questions? No, I think we're good. I don't see any. Anyone?
Great. Well, I really encourage all of you. I mean, I know some of this might seem like a little intimidating, but you know, even if it does, you don't really need any special equipment to go out and just dig a hole in the soil and look at it. And some of the easiest things to do are the root sheathing. Look at how well your roots are coated in soil. Look at your aggregation. Look at biopores in the sides of your hole. Check out your root behavior. Look for topsoil. How deep is your topsoil? How deep do you have to go before it changes to a lighter colored subsoil? These are all some really simple things that you can do to start observing indicators of healthy soil life. And I really encourage you to do it. It's just it's really fun, and you'll just learn so much about your soil.
All right. Well, thank you everyone for joining us tonight, and happy growing.
Very nice series.
Okay. Thank you very much. Thanks, Charlie.
Thank you.
Thank you.